the question that we tried to answer through our research is, can feature selection be used to decrease the amount of data needed to discriminate between different types of major cancers? Our hypothesis to the question was that feature selection can be used to significantly decrease the data required to accurately discriminate between different types of major cancers. The types of cancers that we focus on are breast cancer, kidney renal clear cell carcinoma, lung adenocarcinoma, colon adenocarcinoma, and prostate adenocarcinoma. So cancer is an incurable disease, but it has many treatments such as surgery, chemotherapy, radiation, and cancer immunotherapy. Cancer immunotherapy is a type of therapy that aims to fight cancer by either activating or depressing the immune system. Current research is using machine learning to make cancer immunotherapy more accurate in providing an understanding of the genotype and phenotype relationship of cancer tumors. We used machine learning techniques to identify which genes are most significant in giving rise to specific types of cancer tumors. There are 4 different models that were tested to predict whether or not gene expression values could accurately predict cancer tumors, which effectively clarified that there is a strong relationship between the genes and the type of tumor. Then the random forest feature selection was used to calculate the 50 and then 25 most significant genes in giving rise to a certain type of cancer. Then the 4 models were trained and tested again on the feature selected database to determine if the predictive capability was maintained. We discovered that a highly accurate machine learning classifier can create a small number of significant genes instead of a whole genome. Some cancer statistics that we mentioned in our introduction include: in 2020, there are expected to be 1.8 million new cancer cases diagnosed. It is estimated that 40 out of 100 men will develop cancer in their lifetime, and 39% of women will. And the most common area for cancer is the digestive system. The most important factor in surviving cancer besides early diagnosis is staging. Staging helps indicate how advanced and invasive the cancer is. Cancer is a disease that is driven by genetics, and these genetics control the cells in ways like how quickly the cell grows, how often the cell divides, and how long the cell lives. The genetic makeup of the DNA controls the cells by creating proteins that have specific functions that allow the protein to perform the right function for the cell. When detecting cancer, it is clear that gene mutations do occur, but gene mutations don't always lead to cancer, which increases the need for the understanding of how genes affect the tumors to evaluate the type of tumor. This study uses machine learning, specifically feature selection, to improve the use of cancer immunotherapy. Feature selection is a type of machine learning that improves the accuracy of the model, and it enables the machine learning algorithm to train faster. Feature selection was important to the experiment because it allowed the large model to be understood easily with precision. So in Nath and his team's paper, they performed a survey using data analysis to predict how effective cancer prediction and detection would be using machine learning. They believe that the mix of models and analysis will be very important for cancer detection and research, and that gene expression is a useful tool to predict certain types of cancer, and that continued research will improve the models and advance the classification and analysis to treat cancer patients accurately. Shun and others used 14 methods and gene expression data to create a complex model to be able to detect the type of cancer in a patient. This method had a high detection accuracy, which was around 998%. But to our knowledge, they didn't do a lot of work on feature selection and identifying which genes are most significant in giving a rise to a certain type of cancer. In 2011, Lavanya was able to evaluate the effect of feature selection on classification, specifically with breast cancer, and the results showed that feature selection does lead to more accurate classification, and it increases clinical applicability and data efficiency.
We received our dataset from the University of California Irvine Machine Learning Repository. The dataset was titled Gene Expression Cancer RNA Sequence Dataset. There were a total of 20,531 total attributes in the dataset, and 801 patients were tested. Each patient in the dataset had one of 5 types of cancer: breast cancer, kidney renal clear cell carcinoma, lung adenocarcinoma, colon adenocarcinoma, and prostate adenocarcinoma. In the dataset, each attribute was given a secondary name. These were taken from the RNA sequence Pankin dataset and selected as a separate set to be analyzed. None of the rows in the dataset were deleted because there was no data missing. We split our data randomly, so 70% of the data was used for training and 30% was used for testing. We tested 4 different machine learning methods to see whether or not they could predict cancer and to see if there was a correlation between gene expression and type of cancer tumor. These models were designed and tested using the Python programming language, the Google Collaboratory Development, and the Skylearn library. The first model was a support vector model, which was designed using a polynomial type kernel. The second model was a multinomial logistic regression model, and it was designed to employ no penalty coefficient using a Newton CG solver. Another model we used was the k-nearest neighbors model with the k-value of 5. The last model we used was the XGBoost model designed to use a tree booster with a learning rate of 0.1. To validate each model, we used 4 evaluation metrics. The first metric we used was the precision score, which ranges from 0 to 1. The equation for the precision score was P equals TP over TP plus FP. TP is the number of true positives and FP is the number of false positives. The second method we used was the recall score. The equation for the recall score was R equals TP over TP plus Fn, where TP also means true positive and Fn means false negative. The third method we used was the F1 score. The equation is F1 equals TP over TP plus FP plus Fn over 2, where TP is true positives, FP is false positives, and Fn is false negatives. The last method we used was the accuracy score, where CC represents the absolute number of correct classifications and TC represents the number of total classifications performed. The precision score represents the ability of the model to not classify a patient out as a positive for a type of cancer as negative for that cancer. The recall score, however, represents the ability of the classifier to identify the positive patients for a certain classification. And the F1 score represents the weighted average of the precision score and recall score. The accuracy score is the accuracy in the model in detecting whether or not a patient has a certain type of cancer. We needed to train and evaluate the models again, but this time with a selected number of features instead of all 20,531 genes. In order to identify which genes were most significant, we used a random forest classifier. From these results from the classifier, a list of the most important genes was created. This list represented the most significant to the least significant genes in giving rise to a certain type of cancer. We then decided to create 2 copies of the original dataset, one with the 50 most important genes and one with the 25 most important genes, in order to experiment with feature selection. Then each dataset was divided the same way as the original dataset, 70% designated for training and 30% for testing. Then each dataset was used to train and evaluate all 4 models again. Each model was evaluated using the same metrics as the original 4 models.
So this table, it's basically, to put it simple, to clear up any confusion. It identifies if the model misdiagnosed any patients with the wrong type of cancer, and this affects the evaluation performance because we're going to use the 4 evaluation metrics to get the results. So for example, if we take a look at the support vector machine, which is the first model we used, we see that this model didn't diagnose any patients with the wrong type of cancer. And because all the numbers are in a diagonal line, in this case that's good because the diagnosis side, which is the left side, has all the cancers: breast cancer, kidney, renal, clear, cell carcinoma, lung adenocarcinoma, colon adenocarcinoma, and prostate adenocarcinoma. And that's what the model diagnoses the cancer as. And the top row has what the cancer actually is. So that we see that this model diagnose all the cancers correctly because it's in a diagonal line and they all meet up at a point. And as Madeline explained before, these metrics that support the evaluation metrics are validated to gain a comprehensive assessment of each model. So one means that the model, so if we look at the right graph, the right table, the one means that the model is completely accurate, but like 0.98 and 0.99 scores are also very, very accurate. So overall, all these models during the initial experimentation are really accurate. The reason for the XGBoost evaluations, that's the one all the way at the bottom on the right. Evaluations not all be one is the fact that the model misdiagnosed a few cancers. They misdiagnosed 2 cancers in the initial experimentation. So because, as Madeline also mentioned, because the data set had 20,000 something genes, it would have been really difficult to use all of them. So we chose to select a significantly smaller number of genes to test train the models. So when the model selected the 50 genes most significant in giving rise to cancer, which is called 50 gene selection, they gave the result an evaluation showing here for each model that was tested and trained with the genes. So for example, you can see that most of them were incredibly accurate. There's only a few misdiagnoses for each one, maybe like 2 or 3. So we also did the same process again, except the models were trained and tested by selecting the top 25 most significant genes instead of the top 50 genes. Again, here are the confusion matrices for each model, as well as the evaluation performances. As you can probably notice by the results, the less and less genes we select, the evaluation scores are decreasing and straying away from being as precise and accurate than compared to the initial experiment and the 50 gene selection, but that's expected. But overall, they're very accurate as most are in the 90s range or 1s. As you can see in the XGBoost classification, there are only a couple of diagnoses as same as the other ones because in conclusion, although as you can see that some scores were off by a couple predictors, they were overall accurate since we were able to distinguish and select the top 25 and top 50 genes most important in giving rise to breast cancer, kidney, renal, clear cell carcinoma, lung adenocarcinoma, colon adenocarcinoma, and prostate adenocarcinoma through machine learning models because the results were significant. Because these results were significant, utilizing the machine learning models, this shows that these models can be useful and helpful in future experiments to help learn more and identify more data, patterns, and make decisions that will benefit research, specifically in cancer immunotherapy. These machine learning models can be used to gain more knowledge and extensive data on the gene expressions of these tumors and the significance in how these cases come about so the immune system can battle. And this is our bibliography. These are all the sources we use, like outside sources to gather statistics, and also the data set as well and the reference papers. Thank you. Does anyone have any questions?
Yeah, I had a question actually. What is the difference between your precision score and your accuracy score?
The precision score represents the ability of the model to not classify a patient that is positive for a type of cancer as negative for that cancer, but the accuracy score represents the accuracy of the model in detecting whether or not a patient has a certain type of cancer.
I see. Thank you.